So once again, good day everyone. Today we are going to tackle about the microbiology methods for wastewater water laboratories. So before we are going to proceed, we should consider the critical elements for microbiology. First, we have the personnel, laboratory equipment and supplies, the general laboratory practice, analytical methodology, sample collection, handling and preservation, the quality assurance, records and data reporting, and last, the action response to laboratory results. So here are the materials for total coliforms E. coli. First, we have sterile sample vessels, the colilert media, sterile reagent grade water, the sterile pipettes, vessel rack, incubator, and the UV light. So to start with the methods, after putting the sample through the vessel, shake sample thoroughly at least 25 times. Second, use sterile pipette to draw volume of sample down to 100 ml. Third, tap colilert media snap pack on counter to settle contents. Snap pack open and empty media into sample vessel. That replace cap and shake sample vigorously. Media does not have to be completely dissolved in the same prior to incubation. Place sample in the rack and incubate for at least 24 to 28 hours. After incubating for 24 to 28 hours, the bottles are read using a colilert presence-absence comparator for reference. So the samples that show a yellow color equal to or greater than that of the comparator are total coliform positive. Samples with no color are total coliform negative. So the total coliform positive or the yellow samples and the comparator are checked with UV light. Samples that show fluorescence equal to or greater than that of the comparator are E. coli positive. Samples with no fluorescence are E. coli negative. So after the process, we also have here the total coliform E. coli by quantitray. So this method is used for detection of total coliform bacteria and E. coli in source water and groundwater. This method is approved for the analysis sample of long-term to enhance surface water treatment rule and groundwater rule compliance. So let's start up with its procedure. First, preheat quantitray sealer. Shake sample thoroughly at 25 times. Use sterile pipette to draw volume sample down to 100 ml. Sample may be diluted using sterile deionized water if necessary. Tap colilert media snap pack on counter to settle contents. Snap pack open and empty media into sample bottle. After that, replace cap and mix sample until media dissolves. Media needs to be completely dissolved in the sample prior to pouring into quantitray. Open quantitray by squeezing in at the sides while pulling the foil top away. Pour sample into tray. After that, fit quantitray in rubber insert and slide into the preheated sealer. The quantity tray will go through the sealer and the sealed quantity tray will come out back. Incubate sealed trays at the given degree Celsius for 24 to 28 hours. Then, wells with a yellow color equal to or greater than that of the quantity tray comparator are total coliform positive. Count the number of the positive wells and refer to the provided quantity MPN table to find the total coliform MPN. Once again, good day everyone. I am Kenneth J. Ibay and that's all of my report.
Good morning to everyone. Our assigned topic is all about topic 7, which is microbial ecology. And my assigned report is all about microbial habitat. The habitat is an important concept in biology and microbiology, in particular because microorganisms are greatly affected by where they live. Microbial habitats, including soils, rivers, lakes, oceans, on the surface of living and dead livings, inside other organisms, and man-made structures, and everything in between, provide nutrients and protect cells from harsh conditions. The more places you look for microbes, the more microbes you find. Every environment is stratified, arranged in layers, in terms of the degree of temperature, oxygen, nutrients, and sunlight present. These stratifications make up different niches to which a specific microorganisms or group of microorganisms is uniquely suited. Over the billions of years that microorganisms have been on Earth, they've evolved to fit perfectly almost every niche. They really are everywhere. Microbes live at the depths of the ocean and in the highest clouds. They thrive at extremely high temperatures, near hydrothermal vents, and at extremely low temperatures inside polar sea ice. Microorganisms can be found at extremes of pH, salt, and dryness, but they're also very abundant and ubiquitous at all conditions in between. In addition, some microorganisms are resistant to substances toxic to most life. The microorganisms often use these substances for energy and, in the process, detoxify them. For every system that has been designed to keep out microbes, there is a microbe that has circumvented it. This is because microorganisms are the master of adaptation, and because there are so many different microbes adapted to so many different conditions. What this means is that despite our best efforts, microbes can sometimes be very difficult to get rid of. For instance, the colonies of animals are home to maybe the largest biomass of bacterial cells anywhere, with estimates of 10 cells per gram. However, the number of different bacterial groups are thought to be in the range of 100 or 200. In contrast, the most diverse habitat containing the largest number of bacterial species is probably soil, with somewhere around 500,000 species in a gram of soil. Environments with mixed nutrient sources often have high microbial diversity because many different microbes can grow without one taking over completely. So, the total number of microbes is lower, but the number of different species is higher. In some cases, microorganisms have made their own habitats and grow in large multi-species communities that are obvious to the naked eye. An obvious example of this are microbial mats, which can include many different species of bacteria and archaea. Another slightly different type of microbial community, called a consortium, involves a more intimate relationship between a small number of different microbial species. Two examples of these are lichen and the green sulfur bacterial consortium of marine and freshwater environments. Below are the image of the lichen made up of algae and fungi. The microbial world encompasses most of the phylogenetic diversity on Earth, as all bacteria, all archaea, and most lichenes of the eukarya are microorganisms. Microbes live in every kind of habitat: terrestrial, aquatic, atmospheric, or living hosts, and their presence invariably affects the environment in which they grow. Their diversity enables them to thrive in extremely cold or extremely hot environments. Their diversity also makes them tolerant of any other conditions, such as limited water availability, high salt content, and low oxygen levels. Here is the figure in which microorganisms in a cold environment, which is the ice algae in Antarctica. Another figure is microorganisms in a hot environment, which is algae growing in a hot pool in New Zealand. Not every microbe can survive in all habitats, though each type of microbe has evolved to live within a narrow range of conditions. Although the vast majority of microbial diversity remains undetermined, it is globally understood that the effects of microorganisms on their environment can be beneficial. The beneficial effects of microbes derive from their metabolic activities in the environment, their associations with plants and animals, and from their use in food production and biotechnological processes. In turn, the environment and the recent temperature anomalies play a crucial role in driving changes to the microbial communities. For instance, the assemblage of microbes that exists on the surface of seawater is thought to have undergone tremendous change with respect to composition, abundance, diversity, and virulence as a result of climate driving sea surface warming. For microbiologists, it is critical to study microbial adaptation to different environments and their functions in those environments to understand global microbial diversity, ecology, and evolution. They rely on specific physical and chemical factors such as measuring temperature, pH, and salinity within a certain geography to formulate a comparison among microbial communities and the environment different species can tolerate. That's all for today. Thank you for listening.
Hello everyone, I'm Nel Benemenusa Katayas and I will discuss the microbial habitat and diversity. What is microbial diversity? Microbial diversity can be defined as the range of different kinds of unicellular organisms, bacteria, archaea, protists, and fungi. Various different microbes thrive throughout the biosphere, defining the limits of life and creating conditions conducive for the survival and evolutions of other living beings. So, what are the four types of diversity in biology? So, we have it here the four types of diversity in biology. First is species, second is genetics, third is ecosystem, and lastly is the global biodiversity. First is species diversity. Species diversity refers to the variety of organisms in the environment, and they can be further divided into three. First is species richness, the total number of species in a given region or locality. Next is spatial abundance. This is relative number of organisms among the species, the number of individuals per species. And then the last is taxonomic diversity or phylogenetic. It is the genetic relationship that exists between various groups of species. This kind of a diversity is represented using the hierarchical classification based on the phylogenetic evolution of different species of organisms. Second is the genetic diversity. Genetic diversity encompasses all the variety of different genetic information contained in animal. A single species of organism with diverse genetic diversity portray more adaptability and survival mechanism against adverse environmental conditions compared to organisms of a single species with the same genetic makeup. The third one is the ecosystem diversity. It is relatively similar to the species diversity except for the fact that it deals with the variation in ecosystems within a geographical location. From one region or country to another, there are different ecosystem or biomes with examples such as alpine, meadows, taigas, wetlands, grasslands, mangroves, and deserts among others. Last is the global diversity. Global diversity refers to the range of differences that depicts the composition of the group of two or more species in the global context. According to the International Union for Conservation of Nature or IUCN, there are approximately 1.5 million different species of plants and animals spread across the world. 70% represent animal species while 22% represent plants. So, why is microbial diversity important? It is important because there is the need to maintain ecological processes such as decomposition of organic matter, nutrient cycling, soil aggregation, and controlling pathogens within the ecosystem. That's all of my report. Thank you.
Hello everyone, I am Philgumbi Fernando and I am a sign of nutrient cycle. So a nutrient cycle or illogical recycling is the movement and exchange of inorganic and organic matter back into the production of matter. So the energy flow is an unidirectional and non-cycle pathway, whereas the movement of mineral nutrients is cycling. Mineral cycles includes the main cycling, which is the carbon, water cycle, and the nitrogen cycle. What is the carbon cycle? So ang carbon cycle is the chemical backbone of all life on Earth. All the carbon currently have on Earth is the same amount we always had. Kahit sa bagong buhay, nagpo-form ang carbon sa kanyang molecules and protein to form carbon dioxide. It's also found in atmosphere. But the carbon cycle is nature way reusing carbon atoms travel to atmosphere and back to atmosphere over and over again. Pero karamihan ng carbon ay nare-restored through rock and sediment. And ang iba naman sa oceans, then and also dutong nabubuhay doon sa ocean yung mga living organisms. So like Mars plants to fish, then seaweed and birds. Actually nagproproduce din sila ng carbon. But sometimes the dead body of animals is become the fossil fuel so that it become carbon.
That's all for the carbon cycle. Let's proceed to the water cycle. Earth is a teeming with life. Earth life lives in the oceans and on the land. Without water, all land will be desert and all life on land will be dead. The process by which water is brought to the land is called the water cycle. The water cycle has 6 basic parts. Part 1 is evaporation. The sun's heat turns liquid water in ocean and leaks into water vapor, which rises into the air. Part 2 is transpiration. Sunlight on leaves evaporates water, causing plants to fall groundwater through the air roots. Back into their leaves. As a result, the air in above forest fills with the water vapor. Part 3 is condensation. As water vapor rises higher, it condenses back into liquid drops and forms clouds and fog. Part 4 is precipitation. Water droplet in clouds get so heavy that they fall as rain, sleet, and hail. Part 5 is transportation. Rain and melting snow fall earthward to form waterfalls, rivers, and streams that all flow downhill. Part 6 is collection. All river throughout runs back into the sea. Into lakes and into the ground, where the process begins again. That is the water cycle.
So let's proceed to the nitrogen cycle. So the nitrogen cycle is the refinting cycle of processing during which nitrogen moves through both living and non-living things: the atmosphere, soil, water, plants and animals, and bacteria. So there are 5 stages in the nitrogen cycle, and we will now discuss each of them in turn: fixation or volatilization, mineralization, nitrification, immobilization, and denitrification. So first stage, we need to discuss the nitrogen fixation. Ano nga ba ito? In this stage, nitrogen moves from the earth atmosphere into the soil. Each atmosphere contains a huge pool of nitrogen gas. But this nitrogen is unavailable to plants because the gaseous form cannot be used directly by plants without undergoing a transformation. So, kung may use lang ito ng plants, ang nitrogen gas must be transformed through a process called nitrogen fixation. So, kasi ang fixation, siya yung nag-convert sa nitrogen in the atmosphere into forms the plants absorb through their root system. So, let's discuss the stage 2, which is demineralization. Makikita din naman natin yan sa structures. So, this is the mineralization. Sunod, this stage takes place in soil. Nature moves from organic materials such as manure or plant material to an organic form of nitrogen that plant can use. So, dahil dito, yung nakukontain niya nakukontain ni plants. Dahil sa na-absorb niya through atmosphere, go to soil, and go to yung kuan na yun nalagay na dun sa lupa ay na-absorb yun ni plants. Kaya tatawag yung mineralization. So number 3 is nitrification. The third stage, nitrification, also occur in soil. During nitrification, the ammonia in the soils produced during mineralization is converted into compounds called nitrites and nitrites. Nitrites can be used by plants and animals that can consume the plants. Some bacteria in the soil can turn ammonia into nitrites. Dahil kinakain nila ito. Although nitrites is not usable by plants and animals directly, above the other bacteria can change nitrates into nitrates. Dahil katong kinakain ni another bacteria, yung nitrates, yung bacteria nito yung nitrates naman kinakain ng another bacteria, tinatawag yung nitrates. So number 4, immobilization. The fourth stage of the nitrogen cycle is immobilization, sometimes described as the reverse of mineralization. These two processes together control the amount of nitrogen in soil. Sila yung nagbabalance doon sa pag-process ng nitrogen. Dahil doon, nakokontrol niya yung amount ng nitrogen doon sa soil. So, ibig sabihin lang, just like plants, microorganisms living in the soil require nitrogen as an energy source. Kaya nagkakaroon ng energy source yung plants dahil sa microorganism living in the soil. Then, ang last stage is 5. So, makita itong arrow na ito, papunta doon sa kwan. Ito itatawag na denitrification. In the 5th stage of the nitrogen cycle, nitrogen returns to the air as nitrates. At yung nitrates, yun yung bumabalik doon sa atmosphere para ma-convert sa atmosphere na magiging nitrogen gas by bacteria through the process we call denitrification. Yung tatawag nating denitrification. This result in the all-over loss of nitrogen from soils as the gaseous form of nitrogen moves into the atmosphere back where began our story. So, kita naman natin nga bumabalik siya sa original niya kung saan nagpo-form siya ulit ng nitrogen. So that's all for our discussion. Thank you for your time and God bless.
What is biodegradation? Biodegradation is the process of decay that naturally occurs in the environment due to bacterial and fungal action. Generally, these microorganisms serve as decomposer of the nutrients and soil. Most importantly, decomposition is the process of the nature replenishing ecosystems. Moreover, the large organic molecules are broken down into small organic molecules through enzymes. Definition of biodegradation. Biodegradation is the process by which microorganisms break down organic matters. Depending on the materials, this can take days, weeks, or even centuries. Products that biodegrade quickly are veritable to those that don't. Their harmful contents can be broken down in a shorter period of time. The process of biodegradation is threefold. First, an object undergoes biodeterioration, which is the mechanical weakening of its structure. Then follow biofragmentation, which is the breakdown of materials by microorganisms. And finally, assimilation, which is the incorporation of the old materials into new cells. Why is biodegradation important? Biodegradation helps to minimize the environmental impact of products that appears in the environment, ensuring they decompose into less harmful product, water, biomass, and CO2. It is also helpful from a recyclability standpoint because it allows raw materials to be reclaimed and used more easily at the end of a product's life cycle. What is bioremediation? Bioremediation is a branch of biotechnology that employs the use of living organisms like microbes and bacteria in the removal of contaminants, pollutants, and toxins from soil, water, and other environments. Bioremediation may be used to clean up contaminated groundwater or environmental problems such as oil spills. Bioremediation is a branch of biotechnology that employs the use of living organisms like microbes and bacteria to decontaminate affected areas. It is used in the removal of contaminants, pollutants, and toxins from soil, water, and other environments. Bioremediation is used to clean up oil spills or contaminated groundwater. Bioremediation may be done in situ at the site of the contamination or ex situ away from the site. How bioremediation works. Bioremediation relies on stimulating the growth of certain microbes that utilize contaminants like oil, solvents, and pesticides for sources of food and energy. These microbes convert contaminant into small amounts of water as well as harmless gases like carbon dioxide. Bioremediation requires a combination of the right temperature, nutrients, and food. The absence of these elements may prolong the clean up of contaminants. Conditions that are unfavorable for bioremediation may be improved by adding amendment to the environment, such as molasses, vegetable oil, or simple air. These amendments optimize conditions for microbes to flourish, thereby accelerating the completion of bioremediation process. Advantage of bioremediation. Bioremediation offers numerous advantages over other cleanup methods. By relying solely on natural processes, it minimizes damage to ecosystem. Bioremediation often takes place underground, where amendments and microbes can be pumped in order to clean up contaminants in groundwater and soil. Consequently, bioremediation does not disrupt nearby communities as much as other cleanup methodologies. The bioremediation process creates relatively few harmful byproducts, mainly due to the fact that contaminants and pollutants are converted into water and harmful gases like carbon dioxide. What is the difference between biodegradation and bioremediation? The main difference between biodegradation and bioremediation is that biodegradation is the breakdown of organic matters by microorganisms such as bacteria and fungi, whereas bioremediation is the use of microorganisms to break down environmental pollutants. Biodegradation and bioremediation are two processes of breaking down unwanted materials by the action of microorganisms. However, biodegradation is a natural process while bioremediation is an artificial induced process.
Hi everyone, my name is Junaisa K. Kabuntalan from BSL Science 3, and our topic is all about topic 7, the microbial ecology. And the assigned topic to me that I will be discuss is all about the microbial symbiosis. So what is symbiosis? Symbiosis is generally defined as a condition where two dissimilar organisms live together in an intimate associate that sees both organisms benefit. In biology, the term symbiosis is an evolved interaction or close living relationship and long-term association between organisms from different species, usually with benefits to one or both individuals involved, or cooperating to help each other in order to survive. The relationship in symbiosis could be good, bad, or neutral for either partner. Animal to animal, plant to microbe, and animal to microbe. A symbiotic relationships exist and are well documented. For example, a clownfish hide from predators and among the tentacles and sea anemones. In return, they feed the anemone with their own droppings. The anemone and the clownfish enjoy their symbiotic relationship in biology. And now, let's talk about what is microbial symbiosis. It tends to be a bit broader in definition, being defined as the coexistence of two microorganisms. The partners in symbiosis are called symbionts. The larger organism, termed the host, a symbiotic relationship can be advantageous for both organisms. However, it can also be advantageous, necessary, or even disadvantageous for only one partner. A good host, we as humans also live in symbiosis with organisms. One example is our partnership with bacteria that inhabit our skin and our intestines and help us live more efficiently. 10 times as many bacteria inhabit a human body as there are cells in that body. The vast majority of these bacteria are helpful and sometimes necessary for our survival. You can't see them, but they are microbes. They're on you and you carry more than 100 trillion of them all by yourself. There are three types of microbial symbiosis, and they are categorized as parasitism, commensalism, or mutualism. Also, it can be evident as several different patterns of coexistence. The first pattern is known as parasitism. It produces a benefit to one organism at the expense of the other organism. Parasitism benefits one organism whilst harming the others, and as such is not considered to be a symbiotic relationship between host and microorganism. The parasite is the benefiting organism, and the host is the organism being lived on or in or being harmed. And this can be written as positive all over negative. For example, it's like a classmate exchanging answer sheets without your knowledge during an exam. Thus, they will benefit from the scores, whereas you will be harmed. Also, a good example of parasitism will be the dog and ticks. In biological systems, mainly parasites live or on inside the host body and rely on the host for food and energy requirements, thus exhausting the host resources. The bacteria and viruses that attack us and make us sick are the parasites who are benefiting at the cost of our health. So we are at a loss. And always remember that parasitism is different from predation because parasites do not kill their host. Another type of relationship is called commensalism. Here, the relationship is beneficial to one of the organisms and does no harm to the other. Commensalism is a type of symbiotic relationship where one species benefits whereas the other species remains unaffected, and this can be written as positive all over zero. For example, if your friend will peep into your answers, it won't affect your performance in exam, but it will definitely benefit your friend. Also, a good example for commensalism would be the shark and remora fish. When the shark feeds on small fishes, it might leave leftovers, which is consumed by remora fish. The shark is unaffected by this, however, the remora fish is benefited. And lastly, the relationship known as mutualism. Mutualism, in this relationship, both organisms, bacteria and host benefit. In short, association between organisms of two different species in which each benefits. And this can be written as positive all over positive. For example, just like you and your friend helping each other for an exam, except the fact that you belong to the same species. Also, a good example of mutualism is that the ants and the aphids, who support each other. Ants can transport aphids to new plants to eat and bring them inside the anthill when it is too cold and protect them from predators like ladybugs. Ants do this because aphid poop is basically a sugar paste called honeydew. Ants take care of the aphids because they can then eat the aphids' waste. And that's all. Thank you for listening and God bless.